let's go on to the next presentation. And this will be by Jamie Elsila, who will be talking about organic analysis and lunar samples, sample and curation considerations. And we heard a nice talk yesterday by Danielle Simkus, who sort of laid a nice setup for this presentation. So Jamie, are you there?
I am. Can you hear me?
Yes, I can.
Okay, so I'm really happy to be here. I think this is going to be a great day of talks and discussions. And I want to talk a little bit about organic analysis, what we've learned from past analyses and ongoing analyses, and what that can help us think about for future sample and curation considerations. So next slide, please. So when the first Apollo samples were returned, understanding their organic content was a big question, and there were a lot of studies going on immediately. Included among these organic compounds of interest were amino acids. They were a focus because they're essential to life on Earth, so they're very interesting from an astrobiology point of view. They'd also been detected in carbonaceous meteorites, so they were known to be extraterrestrial compounds, and there were good analytical techniques available to detect them. And in fact, they were detected in the Apollo samples, but there was no consensus on their origins during the Apollo era, and I'm going to talk a little bit more about that in a minute. Other organics were also detected, although some of these analyses were limited due to the analytical and instrumental capabilities at the time. And I've included this abstract from a science paper in 1970. There's a couple of interesting sentences in there. In addition to just pointing out the compounds they observed, such as hydrocarbons, a few amino acids, and related compounds, they discussed the biological controls and the terrestrial rock controls that were analyzed. And this goes a long way towards contamination knowledge, and as Julie was just discussing. And in fact, that last sentence of the abstract says, interpretation of the origin of the carbon compounds requires extreme care because of possible contamination acquired during initial sample processing. And so even back when the first Apollo samples were being returned, contamination and curation was a very important part of making sure that we got the best science knowledge from the analyses, and that continues till today. Next slide, please. So the lunar amino acids were detected at the time, and there was a very lively debate in the literature in the 70s about the sources of the detected amino acids. There was consideration of exogenous delivery, infall of meteorites or interplanetary dust particles to the lunar surface, as well as implantation of precursors by the solar wind. And then there was also the possibility that the compounds that were detected were just terrestrial contamination, either from exhaust from the lunar module propellant or biology, simply because humans were involved in the collecting and processing and curation of the samples. And these different sources can be distinguished by looking at what compounds are present, which structures are there, and how they're distributed, or by looking at variations between samples collected from different environments on the lunar surface, and even by looking at the isotopic signature of the individual compounds. But in the 1970s, this wasn't possible given the technology at the time. But because of sample return and careful curation, re-examination was possible many years later. Next slide, please. And I just want to talk a minute about some of the results that were observed and what lessons those can have for us as we look forward towards the next sampling. A re-examination of lunar samples showed that the amino acids detected, many of them seemed to be primarily from terrestrial contamination. There were a lot of similarities, both isotopically and in terms of which compounds were present, similarities to biology on Earth. But there were amino acids present that are not common in biology and seemed to be contributed from meteoritic infall to the lunar surface. In addition to that observation, it was noted that many of the amino acids were formed from precursors during the laboratory workup method, the extraction and hydrolysis. And Danielle Simkus' talk yesterday spoke about some of these volatile precursors, things like hydrogen cyanide and amines, that may be present in the lunar soil and forming amino acids during analysis. Other observations were that the least mature samples, the ones that had the least exposure to the lunar surface, had higher abundances of organics, suggesting perhaps that there's better preservation in the least mature samples. There was also a lot of heterogeneity observed. We looked at a 10-gram sample of a lunar regolith, and we split it into aliquots of between 0.5 gram and 1 gram. And in those different aliquots, we saw a variation from seeing less than 1 part per billion of amino acids up to 48 parts per billion. And so that tells you something about the heterogeneity and the scale on which there's differences in the organic compound abundances. And finally, as part of this work, we studied some samples that had been stored in a non-curation laboratory in our co-investigator or co-author Everett Gibson's lab for an extended amount of time. And those samples showed a significant amount of organic contamination that really highlighted the importance of proper curation with an eye towards future analyses. Next slide, please. So that was the previous analyses, and now there's ongoing analyses that can tell us more as well. And this is the Apollo Next Generation Sample Analysis, or ANCSA program, that I know we're going to hear about more later this morning. In addition to allowing science exploration through analysis of these samples, there's also curation questions that ANCSA is letting us answer. And so some of the ANCSA samples are lunar regolith samples that have been aliquots of the same material have been stored under standard curation. And so we're hoping to study those and see whether organic content is better preserved under cold curation or not. Similarly, some of the ANCSA samples, there's a sample that was sealed under vacuum on the lunar surface, and it can be compared to samples that were not sealed that way, again, to look at whether vacuum curation, vacuum sealing, is a better way of preserving organics long-term for future generations. Also, in looking at these samples that have been curated under these special conditions for long-term, we can look at signatures of contamination and try to understand how and when terrestrial contamination may have been introduced. And so we're hoping that studying these samples may answer not only science questions we have, but give us information about curation practices that are specifically beneficial or harmful for organic analyses. And unfortunately, with the pandemic, these studies are currently delayed, but we hope that within the next year we'll be able to address some of these questions. Next slide, please. On my last two slides, I just want to talk about what we've learned from these past analyses about sample considerations and also about curation considerations with an eye towards organic analysis. So as with most scientists, I think I'd say we want lots of samples from lots of different environments. We want a variety so that we can really test and understand the distribution of organics and the preservation of organics in different environments on the lunar surface. But some of our previous analyses can give us some clues about how to best look for organics as well. As I said earlier, the lower maturity samples previously had higher amino acid abundances, and so that may tell us that we need some subsurface samples or samples that haven't been exposed to weathering processes as much in order to really look at the organics. We don't yet really know the effects of the shadowed regions versus unshadowed regions on organic content. Some of those ANCSA frozen samples that we plan to analyze were collected in shadowed regions under overhangs from boulders versus sunlit regions. And so we hope to see if that has a noticeable impact on the organic content. I mentioned earlier that heterogeneity on the less than 1 gram scale, and also many of these organic analyses use sample sizes of between 1 and 10 grams. And I think it's important to realize that when determining how much material to sample and to consider allocating for analyses. Though I know we just heard from Julie about what the total limitations will be on the amounts that can be returned. Next slide, please. And this is my final slide. I just wanted to talk a little bit about curation considerations from an organic point of view. And as Julie mentioned earlier with contamination knowledge, witness materials are really key to understanding this potential contamination, especially if we're talking about analyses that might be done generations in the future by scientists who aren't even born yet. If there are materials that can follow the samples around from collection through curation that can then be analyzed in parallel, that gives a really good way of understanding what analytes might have been contributed by contamination and what might be actually indigenous to the lunar samples. I said we don't yet fully know the effects of these non-standard curation practices from the materials. Yep. But we hope to have some answers on that, specifically on how they affect organics in the next year. Material selection and cleanliness is also of key importance for organic analysis, thinking about things that can shed organics or off-gas. And I know that that's something being strongly considered. And I'll just say Teflon is really good for organic analyses. And it's impossible to fully avoid contamination, But with careful thought, like what's going into the planning now, we can really minimize and understand it better. So thank you.
Thank you very much, Jamie. I think it's going to be a whole new ballgame with organic analyses in these very cold and permanently shaded regions. So this is very exciting.